Okay, so last time we talked about cloning, and we went through everything with cloning, how it pretty much involves three organisms, one to donate the nucleus, one to donate the cell, pretty much without the nucleus, and then one to be the surrogate mother to have the cloned baby. So the surrogate mother, none of that mother's DNA goes to producing the clone. It's two other organisms. But anyway, so anytime we're doing these DNA transplantations or nuclear transplantations, because we're putting the nucleus in an empty cell, a lot of times the clone embryos, they either don't develop or they have birth defects. So it's not a really good process. There's a lot of variables involved and a lot of mutations occur, so they don't always work out. All right, so getting away from cloning, there are certain cells called Blastocis, and here, I'll spell it out for you guys, because I don't think it's on this slide. Blastocis. All right, so these are going to develop within 5 days of fertilization. So when you are a brand new baby cell, one cell, within 5 days, you're going to become a blastocyst. So the inner cell mass of this blastocyst, this is going to be the source of all the stem cells capable of forming all the different types of differentiated cells in that embryo. So adult stem cells cannot give rise to all these different cell types in an organism. But these blastocysts, which can develop into embryonic stem cells, they can. So in other words, guys, once these stem cells become adult stem cells, which means they're probably going to have a final origin, a final type of cell they're going to get to, those adult stem cells, they can't give rise to all the different cell types. So here is our stem cell. They still can be a stem cell here. Now these are precursor cells, adult stem cells pretty much. There's only a limited amount of types of cells they can go to. And this adult stem cell here, pretty much, you guys can see that it's in the bone marrow in the middle there. It's pretty much just going to be a blood cell, all right? Where embryonic stem cells can go to a bunch of different ones. So we already talked about pluripotent cells last time. They can become many, many, many different cell types, but they can't be used for cloning to be that embryo because they're not going to be giving rise to an embryo. Researchers are able to reprogram fully differentiated cells like they did for cloning to act like embryonic stem cells using retroviruses. And retroviruses, they can introduce many different types of regulatory genes in order to get it back into an undifferentiated cell. And if you remember, again, this is how we cloned with de-differentiating that nucleus cell. Okay, so these are some new methods, guys. Cells of patients suffering from certain diseases can be reprogrammed and used for testing for potential treatments. So we're talking about like cancer or diseases here. Problem is, it's only for research. It really can't, like this stuff, they'll research it, but for every single person, it's going to be a little bit different. So it's kind of hard to use them for every single person and come up with a specific treatment plan because it's going to be different. Meaning, if they do find out what's going on, it's going to be a whole lot of money to do that treatment. All right, so we're going to go over some abnormal gene regulations here. Two different types of genes. We're going to have oncogenes and proto-oncogenes. Now, I always hated the wording on these. Proto-oncogenes are the good ones. So the one with the extra is a good gene. So the proto is good. Oncogenes, these are bad ones. These are cancer-causing genes. So we do not want oncogenes. We want proto-oncogenes. But proto-oncogenes, these are proteins that normal cell growth, normal division, nothing is crazy there. So we can have some type of mutation, some type of genetic change that occurs. And when that genetic change occurs, we can have a proto-oncogene change into an oncogene. All right. So proto-oncogenes, they code for proteins that are for cell growth. If a proto-oncogene turns into an oncogene, those proteins for cell growth, they might change where the cell grows faster, the cell divides faster, and that's not good. So here's a proto-oncogene. If the promoter gets changed, we can get changed into an oncogene. If we have different control elements, we can get an oncogene, or if we have a mutation in the gene, we can have an oncogene. And sometimes they're okay, sometimes they are not. All right, so if we see here, the promoter, no big deal. The control elements, no big deal. But if there's a change in the gene, this is where we can have a hyperactive protein that can make cell growth increase exponentially. So with our proto-oncogenes, we can convert them into oncogenes, as we already talked about. Now, the second and the third reason here, the second and third bullets, I'm not going to really go over these too much. We can have amplification, which is just increasing our proto-oncogenes over and over again. We can have a point mutation in the gene. That's the third one there. But let's talk about the first one. Movement of the oncogene to a position near an active promoter, which may increase transcription. So let's go back to our chapter on gene expression. So with our first reason, what this is saying is that we have certain control elements and mediator proteins. If you remember, we had the control elements, the mediator proteins, we had the DNA bending, all that kind of thing. And they are both going to be needed to induce the activators to reach the promoter. All right. So if we move the oncogene to a position near the promoter, when that DNA bends, it can actually increase our transcription rate. All right, next one, tumor suppressor genes, and these just function as they sound. Tumor suppressor genes, they are going to be used to suppress tumors, so they're good. We want tumor suppressor genes. These are going to prevent our cells from rapidly dividing, all right, so they're good. The other thing they can do is they can encode proteins that promote DNA repair. So they can repair the DNA if there is a mutation, so that'll kind of not take away cancer, but if there's a mutation that may cause cancer down the road, it could fix it. The other thing it can promote is cell-to-cell adhesion, which is good because then if the cells are adhered to one another, they're like, okay, I'm next to all these cells, I'm surrounded, I'm not going to keep dividing, which is good. And if you remember a long time ago, we went over they have this kind of law they abide by with density dependence, all right, and anchorage dependence. So both of those. So we want the cells to be surrounded by other cells. Two other genes we're going to go over. There is a RAS proto-oncogene and a p53 tumor suppressor gene. So these are ones that are common for human cancers. Mutations in the RAS gene can lead to production of the hyperactive RAS protein, and then what will happen there is the cell just keeps dividing. So here up top we have the RAS gene. Everything is good here. We have a protein that'll tell the cell to go through the cell cycle. Down here we have a mutation in it, and what that's going to do is cause overexpression of that protein, and that's going to cause the cell to go through the cell cycle a lot faster than normal. So instead of doing that 90% interphase as normal, it's going to speed that up. All right. So p53 prevents the cell from passing on mutations due to DNA damage. The p53 gene, it's an activator for a bunch of other genes. So p53 activates a bunch of other genes to get expressed. So if we have a mutation in p53, what this does is it prevents the suppression of the cell cycle, meaning since it's an activator for other genes and saying, okay, now you can form, okay, now you can form. Instead, those other things are going to be forming a lot more if there is a mutation. Okay? All right. So, I think it's the last thing, yeah. Last few slides of the chapter, guys. So this is just going to go over cancer a little bit. And there's two different types of tumors out there. There is benign tumors and malignant tumors. Benign tumors, they're bad, but they're not like real bad. It's just like going to be a growth of cells. It's more localized to one area. It usually does not metastasize, and they do not spread. As you get older, your incidence rate of cancer does increase. Hopefully your cells catch it. That's our goal. We get mutations in our bodies every single day, all right? But more than one mutation is needed for cancer to occur. A bunch of things will cooperate together in order to make sure we do not get cancer. So usually there are proteins that are going to regulate the genes becoming proteins and becoming expressed. So what needs to happen, guys, is the proteins that regulate it, there has to be a mutation in there, and there has to be a mutation in the gene that is turning into a protein. So there has to be a mutation in both, because if there's just a mutation in one, the protein's going to recognize it and be like, hey, okay, we're done here. Let's not make any proteins from this because there's a mutation. All right. So we usually need more than one. So another way to say this is we need an oncogene in there, and we need the loss of the tumor suppressor genes. So we need both of those to go wrong for us to get cancer. Here is, kind of gross, but colon cancer. You can see the tumor there getting bigger and bigger, and this is malignant. If it was benign, it would be kind of here at the beginning. Now, the benign tumor can turn malignant over time, as you can see here. What happened was we had this benign tumor, and then the tumor suppressor gene stopped working, and then it grew, and now it's malignant, and now it can spread. All right. Based on your article reviews, guys, that you're doing, maybe you started them, a lot of them had breast cancer in them, and these were the BRCA genes. So BRCA genes are good. These are tumor suppressor genes that are in women, and they will repair DNA damage that can lead to breast cancer. So that's good. But if there are mutations in these genes, they found that this is how women can get breast cancer. Now, the BRCA1 and BRCA2, these are inherited genes. So you get them from your mom. So if these are passed on, they do increase your incidence for, or percent chance of getting breast cancer. There are, and it says here, they're found at least half of inherited breast cancers. Yeah, they are, but they only account for like 20 to 25% of all breast cancer mutations in these. So don't think just because you might have the mutation that you are going to get breast cancer. All right, last slide here. If DNA breaks, that can cause cancer too. So things like if you go tanning in a tanning bed, this can cause DNA breakage. A bunch of carcinogens, ones found in cigarette smoke, anything like any kind of smoke, any kind of inhalation of foreign things. Chewing tobacco, that has a bunch of carcinogens in it as well. All right. Viruses can also, when they inject their DNA, they can give you an oncogene. And this can disrupt the tumor suppressor genes. And yeah, hopefully it doesn't convert a proto-oncogene into an oncogene, but this does occur from time to time. Okay, guys, so that is the end of our chapter 16. We will have a test day after tomorrow. So just finish up your study guide or reading guide for chapter 16. Have a good rest of your day.